Hello, everyone, and thank you for joining us today. I'd like to welcome all of you to today's presentation on focused ultrasound for temporary opening of the blood-brain barrier and the therapeutic applications of this technique. Today, we'll hear from Dr. Elisa Konofagou, who has been a leader in this field, opening the blood-brain barrier to enable potential treatment of Alzheimer's disease, Parkinson's disease, and other neurological disorders. A quick note for those of you on the webinar: if you'd like to ask a question of Dr. Konafagou, please submit it via the chat function. We will collect these questions until the Q&A period begins at the end of the presentation. And now I'd like to introduce Dr. Elisa Konofagou. She is Professor of Biomedical Engineering and Radiology at Columbia University, where her lab works on developing novel, ultrasound-based techniques for both imaging and therapeutic applications. As I alluded to before, she is one of the world's experts in using focused ultrasound to open the blood-brain barrier to enable drug delivery to the brain for a wide range of clinical uses. And now, Elisa, please take it away.
Thank you very much, Jessica, and it gives me a pleasure to be with everyone over this webinar. I would like to also thank the Foundation for organizing this very swiftly. I will spend the next about 45 to 50 minutes giving an overview of what we've been working on, mainly in my group, for the opening of the blood-brain barrier and then be able to deliver drugs that would treat disease. So the outline of this presentation starts with the definition and motivation behind what we're trying to do, which is opening the blood-brain barrier. I'm going to show you what the blood-brain barrier opening looks like and how we induce it, and then some of the standard questions that we have been trying to answer over the years, such as the safety, the role of the microbubble that's injected, the mechanism, optimizing the parameters that are used in ultrasound, how the blood-brain barrier closes, and then a lot on drug delivery. So how we can deliver neurotrophic factors, especially for neurodegenerative disease treatment, and of course large animals and clinical applications. So we start with the definition and motivation. Our focus is neurodegenerative disease, so that's alluding to the neurons that are slowly dying in the brain, degenerating fast, and of course that includes Alzheimer's disease, Parkinson's disease, which are the biggest diseases in the brain for neurodegenerative diseases, but of course also amyotrophic lateral sclerosis, also known as ALS, and Huntington's disease. And what these diseases have in common is that they actually are affecting specific regions of the brain, and especially very deep regions, subcortical, in the midbrain itself, making it very difficult, even with invasive techniques, to treat. Now, these diseases are on the rise, especially Alzheimer's disease and Parkinson's disease that have logarithmic rise in the population, mainly because of the much better techniques of diagnosis and detection that are happening at earlier stages. So our focus is mainly on opening the blood-brain barrier. This is what it looks like under a scanning electron microscope. So this is a cross-section of the capillaries. You can see that we have the endothelial cell that is wrapping around the lumen where the blood is flowing. And there's different ways that you can cross what's called this barrier from the lumen to the surrounding tissue that you see, and any molecule that's flowing in the circulation that's over 400 Daltons cannot cross this barrier. Now, this barrier has to do with the tight junctions, which are protein strands that are linking the two ends of the endothelial cells. There's also enzymes that can transport through this barrier, and also transport systems. I'm going to get to that in a minute in the next slide. So what is happening with the blood-brain barrier is that it protects the brain from toxic molecules in the blood, and it effectively works very well, preventing 99% of smaller molecules and 100% of very large molecules. So it's great for a healthy brain. And you see a schematic here on the right that shows exactly the same thing that I showed you on the microscope, but also surrounding the astrocytes and some interneurons that are linking the neurons that we want to reach around it. And then, unfortunately, for drug delivery for an unhealthy brain, only 5% of the drugs can currently penetrate. So it has been proclaimed for several years now as the rate limiting factor for brain drug delivery. This is another schematic of how we can cross the barrier. We talked about the tight junction, so this is the endothelium, showing you the endothelial cells in these rectangular blocks, the astrocyte on the other side, and the lumen is on the top. So you have 5 different ways of crossing this barrier, either with this aqueous pathway, with water-soluble agents through the tight junction. Transcellularly, there's a few mechanisms, including the lipophilic pathway, with lipid-solid agents, the transport proteins, and also some receptors that you can link to, and then finally, such as insulin and transferrin can go through. And we're going to talk about that, how we can affect some of these endothelial receptors in the neurons. And then finally, the transcytosis that's actually used by the albumin in plasma proteins. So what can ultrasound do? Ultrasound, and focused ultrasound in particular, relates to a curved source. This is a concave source, very similar to what we do in optics as an optical lens. It has a specific geometric focus that's governed by the geometry of this acoustic transducer or source that sends waves that are concentrated at higher intensity at the focus. The dimensions of this focus are governed, again, by the geometry, such as the diameter and the focal depth, but also by the frequency or wavelength of this wave. So we can vary the transducer properties, and we can vary also the focal size. But the main thing is that this is extracorporeally applicated or non-invasively, and you can have the concentration of the ultrasound waves in a specific region and leaving everything around it unaffected. So it's a non-invasive and localized delivery. I'm going to start talking about the mice. This is what we have done most of our studies on, and then later on I'm going to show more on the monkey side and the clinical applications. So this is what the transducer looks like, much bigger than the mouse head, much bigger than the mouse brain. What we do on the mice is that we shave the fur that's reflecting some of the ultrasound. We target a specific region in the brain. In our case, it will be the hippocampus that relates to Alzheimer's disease, or the caudate putamen and substantia nigra that relate to the Parkinson's disease. We inject microbubbles intravenously, and I'm going to talk about that later on. And we apply focused ultrasound after the microbubble injection. The blood-brain barrier opens instantaneously, and then we inject the molecule of interest intravenously. So that will be either a contrast agent on the MRI that allows us to see where we have opened, or the drug of choice to be able to treat the region. And the molecule deposits through the blood-brain barrier open regions instantaneously. Taking according to the kinetics a few minutes. So what we're trying to do, as I mentioned before, the blood-brain barrier will open non-invasively, selectively in a specific region. You can also apply it through the whole brain in raster scan format. That also has been shown by others. Transient is key, so the blood-brain barrier opening will be reinstated. And then a single application is what most of the results that I'm going to show relate to, but as I said, that can be applied also sequentially in different regions. This is an illustration that we have developed a while back with Columbia University, showing you the ultrasound beam here vertically, the actual vessel, the microbubbles that are flowing, and also the drug in purple. And we want to reach the neuron that's right next door, usually 10 microns away from the vessel. And by opening here the tight junctions and making the vessel oscillate through the oscillation or cavitation of the microbubbles, we allow for the drugs for the first time to cross these tight junctions and finally reach the neuron that we want to treat. So in our case, we have been looking at these horizontal sections of the mouse brain, and we have done a lot on the hippocampus, as I mentioned, that is related to early onset Alzheimer's disease. And we utilize the actual symmetry of the brain. So therefore, the targeted hippocampus is on one hemisphere, and the control hippocampus is on the contralateral hemisphere. After opening, the mouse is introduced in the MRI, and we're able to see where gadolinium was able to permeate. Normally, the gadolinium does not permeate through the barrier. It's much larger than the barrier can allow. It's mainly contained in the vessels and the ventricles. In this case, we actually can see how it permeates and diffuses into the hippocampus, whereas the contralateral side is devoid of the gadolinium. And I'm going to talk also about the closing later on, but this is a view of the next day where we inject exactly the same amount of gadolinium, and in this case, gadolinium does not permeate, showing that the blood-brain barrier has closed. So that was in the hippocampus, and I'm going to talk about also these two regions that are affected by Parkinson's disease, which are the caudate putamen on the top and the substantia nigra on the bottom. And we have a three-dimensional image of the MRI showing you the opening in bright orange, and dark orange is actually the region of the brain that has the blood-brain barrier intact. So therefore, in this case, we have the localized delivery as we want it in the regions where we want to treat. And through other sequences in MRI, such as T2-weighted and diffusion-weighted imaging, we can actually see whether we caused hemorrhage or edema. In this case, we have identified regions and parameters where we would not cause edema, normally showing up as an enhanced gadolinium region. Almost a decade ago, we were able to show this in Alzheimer's mouse models. We wanted to see at the time whether the blood-brain barrier was leaky, and also see whether we were affecting the amyloid plaques. In this case, we sacrificed the mice a day after of the blood-brain barrier opening. We will see the same kinetics of the gadolinium. At 90 minutes, we were able to see permeation through intraperitoneal injection of the gadolinium throughout the surrounding gadolinium region of the hippocampus. And then the next day, again, the same idea that the closing was reinstated. Other groups in Australia have shown that the plaques can actually be removed. This is something after a couple of months. This is something that we were not seeing because this was sacrificed after a day. But this is what the amyloid plaques look like in this model, showing the specks of thioflavin S stained amyloid plaques in the hippocampus of this transgenic model, versus on the right the wild type that's of course devoid of amyloid plaques. So in our case, they were intact. So safety. So how safe is this opening? Us and others have done extensive studies. I'm going to show you some of what we did in both histology and also later on with MRI. So we used the T1-weighted MRI again to show the hippocampal area, and where we show the highest enhancement, we actually guided the histology. You can see on the left where we opened versus the right, there's no difference at 0.3 megapascals. As we increase the pressure at 0.45 megapascals, we also don't see red blood cells except inside the vessels. So that means no red blood cell extravasation in this case. At 0.6 megapascals, however, we do see red blood cell extravasation. And of course, if we go at higher pressures, we also see some dark neurons. So there's a tight window of safety, albeit being a safety window there. So most of the results I'm going to show you are going to be at 0.3 and 0.45 megapascals. We've done also a lot of behavioral studies on mice over several different groups where we sonicated sequentially every week for 6 months, or some controls where we actually didn't do anything to the mice, they just were left alone. And there was virtually no difference in the motor control or rotation on a rotor rod. There was a positive control that we went up to 1.5 megapascals. We knew that we were causing damage, and that was the only one that had abnormal behavior as far as motor control was concerned. I'm going to talk first about the microbubble role and how this affected. We have worked with several different microbubbles, including Optison that we used in early on that I showed you up to now, and Definity, as well as SonarView. In this case, we started a collaboration with Mark Borden almost 10 years ago, and he was able to sort these microbubbles for us and for different diameters. And we realized that when we were doing that, at 1 to 2 micron bubbles versus 4 to 5 microns, we can actually control the size of the opening. So in this case, smaller bubbles lead to smaller opening regions versus larger bubbles that match better the capillary diameter. So it's easier to open with larger bubbles versus smaller bubbles. And in this case, we are operating within the safe regime of 0.3 megapascals. So this also takes me to the mechanism, underlying mechanism. So we operated within the window of safety and also where we actually could see blood-brain barrier opening, and we could see at 0.3 megapascals. And I'm showing you the top panel. It was mainly stable cavitation. This is an ultrasound image. So these microbubbles are also used as contrast agents. And you can see phantom where we opened a channel and we could put microbubbles there, so you can see where the microbubbles are, and we can listen to the cavitation activity, and the microbubbles themselves can actually enlarge or oscillate, as I showed you in the animation, and we can pick up the different harmonics at the fundamental frequency, at the harmonics of the actual bubbles, and also what's called the superharmonics and subharmonic. So this will be within the safe regime. If we go up to 0.9 megapascals, this is mainly governed by inertial cavitation. So in this case, the bubble is not stably oscillating anymore, but it can also collapse. And what you see here at the focus of the transducer is that the bubbles no longer have a nice enhancement, but they in fact are starting to disappear. And you can also pick up this kind of response of inertial cavitation on the actual bubble signature. And on the spectrum itself, you don't anymore just see the harmonics, but you also see what's called the broadband response of the rise of the entire spectrum, showing you that all frequencies of the ultrasound were actually interrogated by the bubbles, including during the collapse. So we're able to actually see that in this phantom, and we're going to show you also later on how we can also do this in vivo and be able to monitor the mechanism in vivo. Now, relating this to the MRI, and I realize this is a very busy slide, so I'm going to just draw your attention on these red squares, and we have divided this with peak negative pressure from 0.15 megapascals, where we see no blood-brain barrier opening, all the way to 0.6 megapascals, where we can see some damage usually. And then we have also sorted it by microbubble diameter. So as I mentioned earlier, at 1 to 2 microns, small bubbles, they need a higher pressure to open. And you also have this associated smaller blood-brain barrier opening volume while staying within the stable cavitation regime. So the stable cavitation regime is actually indicated by a spectrogram, and you see these parallel lines where there's high spacing between these harmonics, indicating that there's very little inertial cavitation. At the larger bubble diameter ranges, including 4 to 5 and 6 to 8, we're able to open larger volumes on the MRI and keeping it under the stable cavitation regime. As we increase the pressure, we enter into the inertial cavitation regime, and you can see more higher bubble activity at different frequencies, indicating inertial cavitation and also larger volumes of opening. Another thing that we've done over the years is actually use MRI and the dynamic contrast to look at the kinetics of gadolinium and be able to map the permeability of the barrier. And again, similar to the previous slide, we changed the pressure from 0.3 to 0.6 megapascals, and then the bubble diameter, and we could see we could go from the top left of no opening at 0.3 with 1 to 2 all the way to bottom right, where we have the largest opening, and also on the right of each MRI gadolinium image, we actually can see the higher permeability that relates to the opening of the barrier. And in this case, we have the largest permeability at the area of the hippocampus. So we can vary the pressure and the microbubble diameter and control both the volume of the opening and also the permeability itself. Some of the recent work that we've been doing is actually seeing what is the mechanism at the cellular level. I'm not going to talk too much about this for the sake of time. This is an arteriole showing leakage. In this case, it's biocetin that has about 1 kilodalton of molecular weight, and we can see this GFP expression on the arterioles and also some leakage around it. And if we compare this to the contralateral side, so what you see is the arteriole, and in green it will be the tight junctions. This is a transgenic model of the mice that express GFP on the tight junctions themselves, and we're able to see that it's completely intact on the contralateral side, and there's only leakage on the side of the ultrasound exposed brain. And the percent of arterioles that we've seen with full leakage is about 47%. Optimization of the barrier itself involves several different parameters. We have looked at the pulse length and the pressure, as I showed you earlier. The pulse length also is very important about keeping the safety and also not radiating the brain with ultrasound redundantly. We've also seen that shorter pulses can open the barrier. This is one region where we actually open with 0.1 milliseconds versus 10 milliseconds that I showed you earlier. And we have the left region of interest here, showing very homogeneous distribution at low pulse cycles versus a more punctated distribution with a higher dose, however, with a pulse length. So this is one of the regions of the hippocampus that we've opened, and one of the parameters of the pulse length that we have worked with. Another one is what's called the burst repetition frequency, so how fast are we sending these trains of pulses versus what is the pulse repetition frequency, so how fast we're sending these pulses. And we found that, and we've published this almost 6 years ago, about the fact that we have 5 hertz for the burst repetition frequency and 100 kilohertz as the best, the optimized sequence for opening the hippocampus and getting the highest dose. But one of the most important things that we also found over the years is that with just 3.5 cycles at 1.5 megahertz, we're able to open with DEFINITY in this case, and again a very homogeneous distribution as we decrease the pulse length versus the contralateral side. When we did actually magnify these results, we will see a more specific distribution of the dextrons that we have used, the 3 kilodalton dextrons at the time, at the regions that we thought initially were capillaries, but then at a further magnification, we found out that they were actually neurons. And I'm going to talk about that later on when we try to target neurons with the neurotrophic factors. Closing itself, that's also a lot of studies that others and us have done over the years. We have also looked at the dependence on the microbubble and the pressure. This is one case where we used large bubbles and low pressures, and we found that it takes in the mice about 2 to 3 days to fully close. And the volume does actually close towards according to the pressure. So we have the highest pressure at the focus, and then at the tails of the focus we have lower pressure, so that closes first. And then finally, day 3, the focus itself at the highest pressures, it has closed back. Of course, the whole story is much more complex. In fact, you can close a barrier within 4 hours, and it depends also on the bubble that is used. In this case, we have seen that at lower pressures, It takes about a day or two to close. At larger pressures, it takes about 3 days to close, and of course that varies with larger bubbles. Larger bubbles are inducing larger volumes, but at the price of actually having this volume, because of the different pressures involved, close much later. This is actually another study that we've done also with Definity and also with a number of cycles, and we've seen that with Definity, the duration of the blood-brain barrier is much shorter, on the order of hours, as opposed to when we use the bubbles that are much larger, that have a larger volume of opening. Again, as I said, with a trade-off of lasting of the closing, having to be reinstated after a few days. So this leads me to the drug delivery part, which we have done also a lot of work on. And so the main question that we were answering in the beginning was whether we can govern the size of the opening. So, and this is some of the work that we did with several different ranges of dextrans that were fluorescent, from 3 kilodaltons, which is on the order of the base or gamma inhibitors for the treatment of Alzheimer's, 70 kilodaltons, which relates to the proteins that are used, including the neurotrophic factors I'm going to show in the next 2 slides, 500 kilodaltons, which is on the order of magnitude of what's used with antibodies, and some of the work that we're also doing in Parkinson's that I won't have time to talk about today. And then 2,000 kilodaltons, which is the molecular weight that mostly is related to adeno-associated vectors, and especially for gene delivery. So what we have seen here is that both for the intensity that relates, fluorescence intensity that relates to the actual concentration of dextrans delivered and the area of the fluorescence, so how big of a spatial distribution of the actual dextrans we can get, it is governed by the pressure. So as we increase the pressure, we can deliver smaller molecules, especially at 0.5 megapascals. But in order to deliver the high molecular weight molecules for the antibodies and the adeno-associated vectors, viral vectors, we will have to increase the pressure and potentially get into the inertial cavitation regime. The same thing holds for the area of fluorescence. Increasing the diffusion of these vectors also is governed by the pressure. I want to say that this was at the pulse length of 10 milliseconds, so we haven't done a study that relates to the pulse length itself, but this is just a pressure study. So the larger molecules that we have been able to deliver is the adeno-associated virus, or AAV, that expresses neurotrophic proteins. In this case, I'm showing you the delivery with the GFP expression, the green fluorescent protein that is actually not a drug, but it shows you what you can expect with the drug itself. And these are coronal sections in the thalamus region, and we're able to see that only in the blood-brain barrier opened region we were able to get this GFP expression. And the contralateral region was completely devoid of any expression. And then we can localize that in the neurons itself. So wherever you see green is actually the GFP expression. Neurons are in red and astrocytes are in blue. And 96% of the expression of the GFP was actually in neurons, but by being able to use a promoter called synapsin that is expressed in neurons themselves. So we can have targeted delivery of the AAV to the neurons themselves. And an even higher magnification, you can see actually the expression on the neurons, where we actually see the soma, the dendrites here of the neuron itself, and then a higher magnification on the right of the colocalization of the GFP expression with the neurons in red. So we know we can reach the neurons. We know we can reach with the highest molecules, and it begs the question whether we were able to actually trigger any molecular pathways that are involved in neuroregeneration or neurorestoration. And one of them is called the neurotrophic signaling, that there are receptors right on the neuron itself. So the actual molecule we have to cross through the blood-brain barrier and reach that receptor. We've worked with GDNF and Nutrien, and what I'm going to also talk about, which is BDNF. So once this receptor is reached, then this phosphorylation pathways are happening inside an expression of specific kinases, such as the ERK1/2 and the MAPK, and then all the way to the actual neuron nucleus that expresses the CREB. So the first thing that we saw 5 years now or more ago was that after sonication of the hippocampus, now what you see here on the left is no longer the gadolinium, it's actually the expression, the localization of the brain-derived neurotrophic factor that's fluorescent, and we're able to see that throughout the hippocampus. And in some of the control hippocampus, which has uptaken the BDNF as well. Now, where does it actually trigger that pathway? This is a MAP kinase pathway activation that you see here in brown, mainly located in the pyramidal neurons of the hippocampus, and we see that also on the unsonicated side, but a lot less. And in fact, if we compare the BDNF injected side compared to the contralateral side, we're able to see that we are able to reach the TrkB receptor into the cytoplasm, expression of MAPK, not so much of the AKT, another pathway for the neurosignaling, and then significant increase in the CREB in the nucleus of the neuron. So with another neurotrophic factor that we worked with Nutrien, I'm going to talk extensively also later on for the treatment that we did in Parkinson's, in Parkinsonian mouse model. In this case, I'm showing you how we can see it in the cytoplasm, stained in brown here, that express the ERK kinase, ERK1/2 kinase, and also here in the nucleus, in the CREB. And if we quantify that, we're able to see that we reach, in this case, the red receptor, ERK1/2, and then into the nucleus. The actual increase was by at least 200%, by at least 150% at the receptor side, and in both the substantia nigra and the caudate putamen. And then on the Nutrien overall, it was on the order of 200% compared to the contralateral side. So now for the actual treatment. So we know we can actually get these neurotrophic factors to trigger neuronal signaling. We wanted to see whether we're able to reinstate any actual neurons themselves. What I'm showing you here is how the nigrostriatal pathway looks like in the MRIs. This is from a diffusion tensor image in a mouse brain. You see that on the bottom are the substantia nigra in green. You have the actual cell bodies of the neurons and some of the dendrites, and then you have the axons that are projecting into the caudate putamen, and there's this arborization of the terminals into the caudate putamen itself. So what happens with the Parkinsonian model that we have used is that what we see here, the dendrites and the axon and the terminals, we inject this toxin called MPTP, and the neuron is degenerating within 5 days. And then we inject the drug through the blood-brain barrier opening, and we hope to reinstate the neuron. So we have done 2 different studies. One is called the neuroprotection, so protect the neuron before the injection of the toxin. So what we do is that we open the blood-brain barrier with 2 different sonications in the striatum and the substantia nigra, and then after a month, we actually deliver the MPTP, it's the toxin itself. We wait 5 days, and we see which neuron survived the toxin. What we have done is that we see no difference between the left, which is the sonicated side, versus the contralateral side. So wherever you see brown on these images are the actual neurons, neuron cell bodies and dendrites in the substantia nigra. And we see no difference between the contralateral side and the sonicated side in any of the groups, except where we open the barrier and deliver these viral vectors that express the GDNF, in this case, the glial-derived neurotrophic factor. And we could see that some of the neurons on the sonicated side were actually kept intact. And then when we quantified that, we were able to see that the ipsilateral side or the sonicated side had significant higher optical density, what you see here in brown, compared to the contralateral side. And that was in the striatum and the substantia nigra, very similar results, that we could see that the only group, including the focused ultrasound only, that we can see the neurons be able to were successfully neuroprotected, were in the blood-brain barrier opened case with viral vectors delivered. So that was the neuroprotection side, which doesn't necessarily make sense in the case of disease. The disease happens first, and then you want to treat it. So we actually increase the stakes here, and we have the study of neurorestoration, where we deliver the toxin first. And what I want to say about the MPTP model is that it does model very well the early onset of Parkinson's disease. Several other groups have done Parkinson's disease. We're certainly not the only one, but they looked at late stages. And the reason why we wanted to see early stages is that this is where it's supposed to be doing the most of the improvement, especially on the motor side aspect of the disease, where the early onset of Parkinson's will be where the maximum benefit for the patient will be. So we deliver the MPTP toxin, again, 5 days for it to induce degeneration of the neurons. And but we also stayed the course for another 21 days. We opened the barrier, validated with MRI, and then we looked at both the protein itself, the nutrient, or the AAV that was expressing nutrient or GDNF. And we did this 3 times every week. We actually we sonicated again and delivered the drug again, and then finally sacrificed the mouse and performed similar kind of staining. So with the AAV itself expressing the glial-derived neurotrophic factor, very similar results with the neuroprotection. We're able to see significance in the ipsilateral sonicated side versus the contralateral, only the case where we open the barrier and deliver the viral vector, and both on the neuronal bodies themselves and the dendrite density that we're able to also measure. And with the protein itself, very similar to neuroprotection, to the AAV as well, we could see that the only group where we could see successful restoration of the neurons, both the cell bodies and in the pars compacta of the dendrites, the substantia nigra pars compacta, we were able to see it only with a group of the blood-brain barrier open with the protein delivery. When we quantified that and separated the dendrites also and the cell bodies, both on the sonicated side on the left and the contralateral side on the right, we were able to find that group D, where we deliver the toxin first with the blood-brain barrier opening and the nutrient, all mice had actually increase of the number of neurons between the contralateral side and the sonicated side. And when we looked at in the neuronal terminals at the striatum, so the other side of the neuron, we were also seeing very similar results with the ultrasound, focused ultrasound on the left versus the contralateral side, and quantification of that showed us that we could increase the treated versus non-treated area increased by 20%. Some other results I want to share with you today will be on some other applications where that could be relevant to some of you. We've shown that you can deliver also non-invasively the viral vectors that are important to optogenetics, such as the expression of channel rhodopsin 2, So what you see on the left is where we opened in the coronal region of the hippocampus here, and it's confirmed by the MRI as the gadolinium is infusing a lot of the brain on the left side. And these viral vectors are more contained in the hippocampus, given that they're several orders of magnitude, up to 5 orders of magnitude larger than the gadolinium itself. But we see a very nice expression over most of the hippocampus with the optovectors. And the larger magnification of that shows you also the neuronal regions and also some of the axons in some of the region of the dorsal region of the hippocampus. Another aspect that we've also published on is on the facilitation of direct injection. There's a lot of direct injection that's been done in the brain, especially for treatment of glioblastoma and other types of disease in the brain. And we were able to see that if we sonicate with ultrasound and permeabilize the tissue before injection, we stand to have 2 or 3 times larger of the diffusion area compared to direct injection itself of this same vector. Another study that we have also published on is about being able to find other routes more closer to the brain. We have been working a lot with intravenous injection and of the drug itself, but there's some published work about potentially reaching faster and higher doses intranasally, and we have found that IN or intranasal injection compares very nicely to intravenous injection of the dextran in this case, and both for the intensity, so the dose, and also the diffusion area, so they compare very well. And we have found that this also translates very nicely to intranasal delivery of brain-derived neurotrophic factors. We're continuing this work and as funded by the NIH to be able to see how much we can actually do this intranasally. And another aspect of the actual vehicle that we're using instead of the microbubbles, through collaboration with Paul Dayton's group, we have used nano droplets. This is the idea being that we can focus the effect even more. Now, these nano droplets are actually made from microbubbles through condensation, and you can revert them to microbubbles just where the ultrasound was exposing the nano droplets. And you can see this high-speed camera video on the bottom right that in the beginning you see nothing, and then as the microbubbles are forming is where the nano droplets are being expanded into microbubbles because of the change in temperature and the change in pressure. Now, the nano droplets are much smaller. They're about 200 nanometers, and it takes and the microbubbles they're converted to microbubbles at the highest pressures. And you can see that this gives us a much more diffuse and homogeneous distribution compared to what we've seen with microbubbles at the same pressures. And also the same thing with H&E and histology for safety. We see that we can extend the safety profile with nano droplets to beyond 0.6 megapascals compared to microbubbles. So the last part of the remaining time I want to spend on the large animal feasibility that we've been doing over the past several years, obviously leading to a clinical. We worked a lot with non-human primates and rhesus monkeys, especially, and also with vesicularis monkeys. And this is what our setup looks like. We have very much based on the mouse setup, and we have the stereotactic apparatus on the monkey itself. And with coupling media from the transducer, now we drop the frequency to 500 kilohertz to be able to penetrate the much thicker skull. This is what a system looks like. The monkey's under this sheet, and we can see the transducer that's lying on its head, suspended by the stereotactic apparatus. The main thing that we always do in the monkeys in real time is monitor the cavitation itself, both for safety and also efficacy. So what you see here is actually the initial baseline, followed by a rapid rise where the macrobubbles reach the region of sonication, and then a plateau of the stable cavitation in blue. Ultraharmonics that lead us only to macro bubble cavitation only, and the flat line of the black line here is the inertial cavitation, where we actually want to keep as flat as possible. That shows us that we haven't entered the inertial cavitation regime, but we're able to say that this is safe during application. So we do this in real time, and we're able to see that this correlates very nicely with MRI. So this are some other examples of this real-time cavitation curve, where we see the rise time of the harmonic stable cavitation, flat black lines, and also some of the activity in red of the ultraharmonics in this region of the putamen on the top, and then caudate in the bottom in the monkey. What we've also been doing over the past few years is also spatially map the cavitation, so where it's actually coming from, by coupling it with what we call the arrayed PCD, or passive cavitation device, which is actually an ultrasound imaging transducer. And we're able to see where the actual cavitation is located also in real time, and that correlates very well with also what we see subsequently in the MR. And this is some of other examples where we see blood-brain barrier opening in the region where our cavitation map has predicted that. So two things also that we wanted to compare with the mice is whether we can have enough gadolinium diffusing, and also the safety aspect. So some of the work we've been doing is also looking at the permeability of the barrier in the monkey, and also quantifying the concentration of gadolinium. So here you see two different examples where we actually do use T1 weighted maps for the blood-brain barrier opening detection in the non-human primates. But unlike in the mouse, where it has a very homogeneous brain, the monkey brain is actually very inhomogeneous, and it makes it a little bit harder to actually pinpoint exactly where the opening happened. So with our techniques that we have published recently, we have the T1 relaxation time, and we're able to see the regions of blood-brain barrier opening, in this case, two different regions in the caudate and the putamen, but also look at the concentration of the gadolinium in these regions, as you can see how it increases by several-fold in the region where it opened. This is in one animal. This is another case where we have opened, again, the caudate and the putamen at the pressures of 0.3 megapascals. And we can also see here gadolinium concentration and the permeability map itself. Now notice that the permeability map in the regions where we open sometimes get interrupted. We were actually very puzzled by that. This is something that we never saw in the mice. And then we realized that this actually has to do with the white matter that has lower permeability than the gray matter. And I'm going to show you a quantification of that in a minute. But this is another example of a single sonication, in this case in the caudate, at larger, higher pressures, where we were able to also see two different openings here, also interrupted by a region that again coincided with the white matter that we saw on the susceptibility to weighted imaging. Another example was how with the pressure we can vary the volume. This also translates from the mice to the monkeys very nicely as well. This is doubling the pressure and also changing the actual region, putamen, caudate, also relevant to Parkinson's disease. And we can see that we can increase both the concentration of the gadolinium and the region of opening just by doubling the pressure. And what I alluded to earlier was the gray and white matter significance. So as we increase the pressure, the peak negative pressure, the volume does increase very nicely in the gray matter and the white matter, but the white matter has a lower volume of opening. This is just two cases, or two different animals, where we showed very similar increase, but much more moderate in the white matter. This is because the white matter is not as vascular as the gray matter. And on the side of the concentration and the delivery efficiency also increases with the pressure. The amount delivered in the case of the monkeys went from 3 micrograms to 20 micrograms as we doubled the pressure. And then the same thing almost also doubled the pressure here in the second animal and doubled the amount delivered, and the delivery efficiency also increased. Safety-wise, we have found that there's also a safety window within which we can operate. Now, the pressures can be lowered in the monkeys compared to the mice with successful blood-brain opening. That's because the pressure is 3 times lower. Therefore, the cavitation is much more likely. And T2-weighted images show you whether any edema happened. Susceptibility-weighted imaging shows you whether any hemorrhage has happened. T2-weighted edema shows up as a bright enhanced pixels, and hemorrhage shows up as dark regions. And within these pressures, in several different monkeys now, with repeated sonication, sometimes on a biweekly basis, we have found that this is pretty safe. Physiological monitoring, we have also published on that, where we see whether the monkeys have changed during sonication or after sonication, the heart rate or the breathing rate, and we have found overall that there's no change in the heart or respiration rate during sonication or even after sonication in both sedate and awake animals. Regarding the closing timeline, we have also found that very similar, it takes a couple of days at the highest pressures, and it can be shorter at lower pressures. This is an example where you see in blue the initial opening region, and then that subsides over a couple of days. Recent studies, as we are nearing towards clinical, we wanted to see whether there's any difference between awake and sedate animals. And now in this case, we have mounted a plate on this animal's head, very similar to what they do in neuroscience for behavioral testing. And we're able to see actually a larger region in the awake animal compared to the sedate animal on the MRI itself. We couldn't relate this to the stable cavitation or inertial cavitation dose, as there was no significance. But we have now hypothesized that it's actually related to the actual vessel size. It's known that vessel dilation is what ensues typically anesthesia, so it may be a little bit more difficult to open larger regions in the sedate animal compared to awake. So we envision that for awake subjects, we may have to lower even more the pressure. And finally, a couple of slides on the future. We are looking into, and we are using now neuronavigation to help guide our targeting even further, and compare it also to stereotactic mode. You see here on the bottom in real time as we move the actual transducer arm and also do away with a stereotactic apparatus altogether, we're able to see here on the crosshairs how well we're doing our targeting. So in red is actually where our beam is head on, and we want to, and in the crosshairs is where we want to target, and it gives us an idea as we move the transducer around how well we're doing relative to our targeting. And at the same time, we are using the cavitation mapping that I mentioned earlier to be able to monitor this in real time. So this is a visual of what it looks like in the MRI itself. After the opening, we see a blue region and very close to where we actually targeted, which is the red circle. So we have very nice targeting on the order of 2 millimeters. And then on the right, you see the cavitation map of the region where we opened, and that correlates very nicely with on the left where the blood-brain barrier opening was confirmed by MRI after the fact. And the final slide I want to share with you is basically going into the clinical feasibility. and detecting cavitation is actually one of the key aspects that we're exploring in our group. We see that without skull, we can clearly be able to detect sonication. I've shown you a lot of the examples in vivo on the monkeys where we actually can detect very nicely what is in red here at very different pressures with harmonic stable cavitation and also the inertial cavitation itself. It gets more challenging when you go through these very homogeneous skulls, such as in the human, where we are seeing significant in some pressures here, which are relevant to us for safety, especially with the stable cavitation detection that we can do through the skull. Now, in the case of the human skull, you have 34 dBs reduction, so much higher in dB than the monkey skull. However, it gets challenging with 500 kilohertz. So in the case of the humans, for efficient cavitation monitoring, we envision dropping the frequency even more. So in summary, I hope I've shown you some of the key aspects of blood-brain barrier opening. The mechanism is that with stable bubble oscillation, we can cause blood-brain barrier opening, which has a higher safety, and we can trigger downstream signaling. We can also open with inertial cavitation. However, that gets more risky for damage. Safety will be for 1.5 megapascals at or below 0.45 megapascals. At 0.45 megahertz will be at or below 0.45 megapascals. The efficacy itself, the permeability, does increase several folds with pressure and microbubble diameter, and we can have some control over that, including the actual volume of opening. For drug delivery, we have looked at mainly at neurotrophic molecules and viral vectors that have shown they can be safely delivered, triggering those pathways. And then for therapeutic effects, we looked at neuroprotection, neurorestoration in the Parkinsonian model. And others, of course, have looked at the Alzheimer's models that, as I mentioned, with amyloid plaque clearance. And for primate feasibility, to our pleasant surprise, the mouse findings do translate very nicely to primates, especially for safety and blood-brain barrier opening volumes and permeability, as well as drug concentration increase as evidenced by the model drug of gadolinium. And finally, for human feasibility, of course, there has been other clinical studies that have been done in France for glioblastoma treatment, and also in Canada at Sunnybrook for also glioblastoma treatment and Alzheimer's. Very exciting results right now that are using the blood-brain barrier opening for treatment. For us, we are developing this transcranial cavitation monitoring that I mentioned, together with neuronavigation in awake subjects, and we hope to start studies later this year or early next year for Alzheimer's patients. I want to thank my group, several of these people that have done all this great work over more than a decade. And at Columbia University, we have several good groups, very complementary on biology and development of transgenic models, and also on the clinical side. And of course, the funding from NIH, NSF, and of course the Focus Ultrasound Foundation and the Kinetics Foundation. And I want to thank you for your attention and take any questions you may have.
Thank you so much, Elisa. And we do have some questions. So the first question, how is the pressure actually measured in vivo, in particular if the wave has to pass through the skull?
That's an excellent question. You cannot measure the pressure in vivo. What we have been doing is that we take the skulls that I have mentioned here in this slide, and we place a hydrophone where you see these regions in blue, so I didn't have the time to mention this. The blue is a putamen, caudate is in yellow, hippocampus will be in red. So we place this hydrophone there, and we try and we measure the pressure there. You can do this in vitro with skulls, or there's really good simulation tools that we also use based on the CT, so computed tomography of the skulls themselves, and you can predict how the wave is going to penetrate through the skull. And we've published on these results. If feel free to reach me and to contact me for if you need guidance in that realm. But you can see the reflection of the skull, and also how the wave, whether the wave can be focused, because some of the pressures could be too high. Some of the frequencies can be also too high, and you can cause aberration through the skull, as well as standing waves on the other side, because the skull also has the other side where the jawbone is, for example, and also the occipital region.
Okay. The next question, well, it's a couple of questions. What would be the best method in your experience to separate bubbles by size without affecting the properties of the bubbles?
So we have used a single method for that. I don't think we're the experts in that. We have gone by what Mark Borden has implemented a few years back, which is based on centrifuge and the larger bubbles floating on top. I believe that what Mark and others have actually concluded, compared to some other microfluidic approaches, that the highest yield will be with the centrifuge approach. However, I'm not an expert in that field, so I can't answer this question as well as I would like, but I can direct, if there's any more questions like that, I can direct to the right people.
The next question, in your neutron studies, what is the time scale involved there in terms of, I guess, in terms of seeing effects, or in terms of the delivery itself? They ask days, weeks, months?
Yeah, so the neutron study, we had, we actually did deliver, in the neurorestoration study, we delivered, I believe, after a month of the MPTP toxin, and we actually did 3 different applications weekly, where we opened the barrier, injected the neutron, and then we survived another week, opened the barrier, injected the neutron, and so on and so forth. So we actually sacrificed the mice after 3 weeks. And that's the maximum we have done. So what I have reported here is actually after 3 weeks. We haven't done any other studies to look at long-term effects to see whether the same effect will last longer. So that's the only timeline that we have worked with.
Next question, is the molecular weight the most important factor for determining whether a molecule will cross the BBB? What about the charge or lipophilicity of the molecule?
Yeah, that's a very good question too. We hypothesized that molecular weight will be the largest. I mean, that will play the biggest role. Now, whether that's the most important thing, we're not sure. But this is the only thing that we have studied. The main thing being that we can study this with dextrans. We have looked at dextrans that have different charges as well. We haven't seen much of a difference, but we have by no means done a study with different charges, but that would hydrophobicity also will affect definitely the kinetics. But the main question that we were trying to answer is whether whether this technology is relevant across the full range of drugs. And one thing that distinguishes these drugs is the molecular weight. But it will be very interesting to see how the charges as well affect it. But very preliminary results that we have done with different proteins, ranging from albumin and also polylysine as well with different charges. We haven't seen much of a difference, but we haven't done a full-scale study on that.
Okay, the next question. Following the opening of the BBB, is there any immune system activity that's attributed to that intervention?
Oh, yes. So I didn't talk about that much, but us and others have looked at several aspects of that, including the microglia that are definitely activated by, during blood-brain barrier opening. In fact, it was attributed to the microglia themselves that would clear the amyloid plaques in the study by Gertz et al. From Australia. And we've also seen, and also some other groups have reported on macrophages as well that are also triggered during that opening. So what we've seen is that some of the aspects of that depend on the pressure and the pulse length, and also there is a reversibility with that, similar to the opening, that it closes back. So this inflammation does also subside according to specific pressures, and as long as you operate within a specific range. But of course, if you increase the pressure or you're doing repeated sonications, other groups have shown that you can actually enter into an inflammation that cannot be reversed.
So I don't know if you are familiar with the maybe a couple of years ago the paper from the UVA group, Jonathan Kipnis, about the discovery of the brain lymph system, and if you could say how that may relate to the BBB opening and potential mechanisms of treatment.
I'm sorry to say that I'm not familiar with that. So if you can tell me something more, then maybe I can answer.
We can have the individual who submitted that question maybe follow up with you with the paper and a question if they still have that, if they still have it.
Sure, that will be fine. Yes.
The next question: does the substance or the molecule, the drug, stay in the target volume or in the brain after crossing the blood-brain barrier, or does it spread farther from your original target due to a concentration gradient?
So this is something we have not looked at, but this is an excellent question. How far does the actual molecule diffuse? We have seen that the gadolinium is contained pretty well after, within the region of interest after an hour, because we perform intraperitoneal injections in that case. However, with the drugs themselves, we have not done a study to see how the diffusion varies over time. The only thing I would say is that it does actually seem to be a barrier also between regions. For example, when we do sonicate in the caudate putamen, we see that the nutrient, for example, was contained in the caudate putamen itself, and it did not diffuse to the cortex. And in fact, there's a very clear boundary, and sometimes enhanced by the staining that we use for the nutrient, showing that you do have some kind of a containment there of the nutrient not escaping from that region. We're not familiar with that separation between different structures in the brain, but that's something that we have seen with the proteins that they are contained within the region of the sonication.
Switching to sort of a more clinical question, in your estimate, how far away do you think we are from routine clinical application of BBB opening based focused ultrasound therapies?
That's it is a very difficult question to answer. There's two questions that we still need to make sure that we answer very, very carefully. One is safety. Are we opening the barrier in humans safely? And this is something that I think all the studies that are currently ongoing or planned in the very near future, safety is number one. So once we show that, then the bar gets even higher. So whether we can treat the disease itself. But I think once we show safety, then I think it will be, because of the need currently in the clinic of so many different drugs that have been developed over the years that could be enhanced in their diffusion and the dose. I think if we show safety, more clinical entities are bound to slowly but surely adopt it.
For the mechanism of BBB opening, are the physical properties of the ultrasound beam, like the shape and the size, just as equally important as the bubble size and the pressure, or are the beam properties less important factors?
These are two different parameters. The beam properties themselves, so the beam width or the focal spot size, will determine spatially where you have opened, and then the pressure and the microbubble size will determine the dose. So how much of the drug will permeate. So the size of where you've opened, how big the size of the blood-brain barrier is. Now, something that's different from thermal ablation is that within that focal spot, you have variation of the pressure. So therefore, the highest pressure will be at the peak of, let's say, this Gaussian, and then towards the tails, there would not may not be an opening, especially if you're operating near the threshold of the opening, where we want to do for the safety aspect, to maintain the safety aspects. So the region where you open is going to be even smaller than what you can predict geometrically by the beam size. So the answer is that the pressure and the microbubble size are important for the drug delivery aspects, because it will better predict where you have opened the barrier and how well the drug could potentially diffuse through it.
We have one more question about the potential of viruses to cross through the BBB. I know you talked about using viral vectors as potential therapies, but I think in terms of safety, this question is getting at, does the opening of the BBB expose the brain to infections?
So we try to address this with what I showed earlier with the colocalization of the GFP expression and the neurons. So the technology of gene delivery over the past 15 years has definitely made a lot of progress, if not has completely changed the field, mainly because first they can engineer these vectors to make them much more targeted by using promoters that are going to target specific cells. So in the case, for example, of the work that we've done for GFP delivery, 96% of the targeted cells in the brain were neurons, and the rest were astrocytes. And in fact, we harvested all the organs of the or main organs of the mouse, including the heart, the bladder, the liver, obviously, and there was no expression of GFP. So we definitely using gene delivery, there's a higher risk because you're exposing these vectors, these viral vectors, to the whole body. But again, with these AAV vectors being much more targeted, we believe that the dose that you increase will be in the cells that you target. And there's a school of thought out there that the gene delivery will be much more efficacious than the protein delivery. There's some work out there with a brain-derived neurotrophic factor for Alzheimer's disease, where they found that the injection of the neurotrophic factor would have to be performed on the 6-month basis or even yearly basis. And there's the gene delivery paradigm is that once you transduce the neurons to be able to make their own neurotrophic factor, you will not need to treat them again. So there's high risk, high gain with viral vectors that's not as much there in the case of the proteins.
Another question, when opening the BBB, molecules are able to enter the brain, does the reverse happen? Do molecules or things in the brain move back into the bloodstream?
So this is a question that we have not addressed in vivo per se. The permeability model that we use, that I didn't have time to describe today, but we have published on that, is the Topf-Kermode model. It's the 2-compartment model where it actually looks at both the K-trans, so this is the permeability that we've been able to map, and also the KEP, so that means the reverse permeability. So how does what is the constant, the permeability constant in reverse, coming from the tissue into the lumen? And what we have found based on the kinetics of the gadolinium is that the KEP is very small, on the order of 10 to the minus 15. Now, this involves, of course, this hypothesis that you have 2 compartments. Basically, one compartment is the blood compartment and the other one is a tissue compartment. And we have completely ignored what happens on the vein side, on the venous side. So obviously the clearance of this gadolinium and all the other drugs will happen on the venous side, and that model doesn't take that into account. However, what it does take into account is that, if you happen to look at the reverse permeability on the capillary side or on the arterial side, it doesn't seem that it compares with the other permeability, the one that goes from the lumen to the tissue.
Okay, thank you, Elisa. Those are all the questions that we have today. So this concludes today's webinar, and I thank you all for joining us, and stay tuned to our newsletter and our website for invitations to future webinars, as well as letting you know that there's a recording of this webinar that will be placed on our website. Thank you so much. Bye.
Thank you.